Hey guys, I'm back with a little bit about cell cycle regulation. And as you can see down here, this is the processes of mitosis over on your left, creating two cells. And this is the whitefish blastula we actually saw an animal cell actually going on. Now, when we talk about cell cycle regulation, there are checkpoints along the way which either tell the cell cycle to keep on going because everything looks good, or it stops. It's kind of like a quality control. And basically, they make sure everything is going along, that the key cellular processes are actually happening like they should, or it stops the process entirely. Now, there are basically three major checkpoints: the G1 and S, the G2 and M, and the spindle checkpoint. The G1 and S are between those two phases, and it basically checks to make sure DNA synthesis is ready to begin. Is everything there? Is it ready to begin? The G2 and M is between the G2 and the mitotic phase, when it actually goes through mitosis. And this makes sure that DNA synthesis went correct. You had the right DNA be made, and it's a full commitment that, yes, we're going to do mitosis. And then the spindle point checkpoint is the last step, and it's during mitosis, and it just makes sure that spindles have attached correctly, and if they haven't, it stops it there and makes sure sister chromosomes or chromatids separated correctly. Now let's look at each one of these a little bit more. The G1 and S checkpoint is probably the primary decision point. It's the one that either gives the go signal or the stop signal. Now, if it gives a go signal, it's either caused by internal clues, such as cell growth may cause it to hit this go signal, or cell nutrition. Is it malnourished, or is it healthy enough to go through the process of mitosis? External signals are like growth factors, are telling it to continue to grow. If it does not receive the go signal, then what it does, it will actually make the cell not divide. Division stops, and the cell goes back into what's called a GO phase, or simply the working state, the maintenance state of the cell. Now, if it got the go-ahead signal, then you're going to get what's called promoting factors, which are going to cause it to go on, or you're going to have growth factors, which stimulate to continue to grow. Now, we don't fully understand this process yet, but we do know that the primary mechanism of controlling this is these CDKs over here, these cyclin-dependent kinases, which are involved in phosphorylation. They're kinase enzymes, and they either activate or inactivate the cell signal, either causing it to keep going or not. And if you look here, the cell cycle signals are like your cyclins, your CDKs, and your CDK cyclin complex, which is represented on the upper right up here, how they hook together. And this basically either tells the process to keep going or tells it to stop. Now, externally, there are some signals that let you know, do we need to continue to grow or not. The one that really sticks out in my mind is the density-dependent inhibition. I would call contact inhibition. You know, most cells, whenever they come in contact with another cell, they stop growing. All right, that prevents the crowding. You know, the crowding is what stops it. So you get this just a sheet. You don't end up with this mass, this tumor, if you will. And there's also an anchorage dependent. It actually has to be anchored into the substrate, a touch sensory mechanism, in order to be able to go. Now, these are with normal cells. Now, keep that in mind, because if we have something go wrong, we could have what's called cancer. And this is what in the proto-oncogenes, if a cell division is activated and it becomes an oncogene, then it's oncogene means it's cancer causing when it's mutated. It switches on and it causes cancer. So, you know, what happens is these cells continue to grow and they've lost some of that density-dependent inhibition that they had earlier. And you could also have tumor suppression gene is actually switched off. That means that the gene that normally causes the cell to stop dividing, if it's cut off, then the cell continues to divide. The example of this is P53, and I want to go into a little bit of P53 because all cancers are going to have the shutdown of P53 in order to be to occur. And what happens is that P53 actually plays a part in the G1 to S restriction point. And what happens is P53 usually will halt cell division if it detects a DNA breakdown. It may cause the cell to apoptosis, which means it causes it to destroy itself. It may cause it to stay in G1. It may make it to go in GO phase, which goes back to that resting stage, or it might fix the DNA. But if you cut off that P53 and it stops it, then the cell just continues going no matter what's there. Arnold Levine discovered this, and it's become a real key component into how we fight cancers. Now, if you talk about cancer in particular, there are 6 things that have to happen for cancer to occur. It's kind of crazy, about 6 things. You have to have unlimited growth, usually caused by cutting on a promoter gene. You have to ignore the checkpoint, so you cut off like the p53 gene. You got to escape apoptosis, which is the suicide gene, so you got to cut it off. You have immortality. You have unlimited cell divisions, so you have to cut on chromosome maintenance gene to make it divide over and over again. You got to cut on blood vessels so you can supply blood flow to this mass of cells you're creating. And you got to cut off the anchoring gene so it doesn't anchor. So you end up with this mass of cells. Now, what could cause a mutation to occur in this p53 or any other gene here? Well, there's a lot of different triggers. UV radiation, going out in the sun, chemical exposure, radiation, pollution, your age could do it, chemicals such as smoking, genetics. Any of these could cause it. And when we talk about tumors or these cancers that are being created, there's 2 types. Don't make sure you know the types. Benign and malignant. The main difference is benign, stay right there where it is, and it usually has some type of lump. So that's when you feel a lump. That's a benign tumor. They're usually not too bad because if we find them soon enough, we can remove them and they're gone. Malignant tumors are the one that's worrisome. It may start in my knee and spread throughout my entire body via the bloodstream. So these malignant ones are not good. Malignants are spread throughout the entire body, and they can attack all your organs and make the organs where they don't work. Now, we do have a couple of treatments for cancers. We use high-energy radiation. It kills the cells rapidly, but realize it kills all cells. So that's not necessarily good. Chemotherapy is a way in which we get the drug chemo, and it stops DNA replication, stops mitosis, stops blood vessel growth, and it kills the bad ones. We hope it doesn't kill enough of the good ones that we don't recover. Chemotherapy, a lot of times people who go through chemotherapy end up getting sick with something else because they suppress their immune system so much. Now, there are drugs that are currently on the market where they're targeting protein enzymes that cause cancer, and hopefully they're going to become more and more prevalent and lead to treatment. Well, anyway, I hope this helps you a little bit with mitosis, how it's regulated, and I will talk to you soon.